Welcome all of you. Today we'll be discussing the first chapter on biotechnology. What is biotechnology? A technology where we are all using living things or enzymes from that organisms. That technology is called biotechnology. Nowadays, biotechnology is used for getting some service or getting some products using these living things or enzymes in bulk quantities, in large quantities. We may be using cells like yeast in making some food products, etc. at home, but that is only in a limited scale. Biotechnology is essentially getting these products done in a commercial scale, on a large scale. Formerly, we used to use an entire organism. But as time changed, as technology progressed, we need use only a part of that organism. Sometimes even few cells or one cell or even a part of a cell for getting the desired product or service. So, what is the correct definition? The correct definition of biotechnology was given by European Federation of Biotechnology. This European Federation of Biotechnology defines biotechnology as that encompasses both traditional view, that is our old traditional view on biotechnology, and modern molecular biotechnology. That definition is as follows. It is the integration of natural science and organisms, either a whole organism or cells or part of that cell, part thereof, and molecular analogs. Sometimes we will not be using original cell products or cell parts. We can use artificially produced, synthesized molecular analogs for products and services. So the integration of natural science, that is biology, and organism cells, part of that cells or that organisms, and synthetic molecules, molecular analogs for getting some product and some services. That part or that branch is called the biotechnology. What paved the way for the birth of modern biotechnology? There are two important reasons. Genetic engineering. What is genetic engineering? Engineering with the genes. That is what is called genetic engineering. What is engineering? Deliberately bringing changes. You are all bringing changes purposefully so as to have a desired result. That type of change is called engineering. So you are engineering with the genes. You are bringing deliberate changes on the genetic makeup. Okay. To introduce these into host organisms and thus change the phenotype of the host organism. The changed genotype are introduced to the organism so that when the genes are changed, their character will be also changed. So you can bring about a desired change in the character of the organism. That's the result of genetic engineering. So we made a lot of progress in genetic engineering, and that is one of the reasons that biotechnology has developed very fast. Second one, bioprocess engineering. That is engineering is the processing of living products. See, we are all getting some service or products, and how you are all getting it? By some process. And that process can be, again, correctly engineered. Maintenance of a sterile ambience, meaning you can now culture a cell or an organism in a cultural medium without being contaminated with microbes. That is, they can be grown for any time in a sterile environment. That is what is called maintenance of sterile ambience. So that is with the help of progress we made on chemical engineering. We have now successfully produced the cultural nutrients or cultural medium in which you can grow cells, and these organisms will be, or the cells will be growing, dividing in their optimum, maximum rate. That technology developed because of the progress we made in chemical engineering. So that to enable growth of only the desired microbe or eukaryotic cell in large quantities for the manufacture of biotechnological products like antibiotics, vaccines, enzymes, etc. Why you are all culturing? You are culturing the microbe or a eukaryotic cell which are genetically modified, so that you will be getting the products. What are the products that you want? You may need some antibiotics for therapeutic uses, or vaccines, or enzymes. These can be produced in large volumes. Okay, so you can culture them in big quantities, and products can be obtained in large volumes. That is the bioprocess engineering. So processing of that genetically modified organisms or cells, so that you will be getting the products in large quantities. We made a lot of progress in these two fields. It is because of the progress we made in these two fields, now biotechnology is a very, very advanced branch of biology. Now, what actually is the advantage of biotechnology? Let us see. Sexual reproduction is having an advantage over vegetative or asexual reproduction, because in sexual reproduction there is the involvement of sex cells, and when the sex cells are formed there will be genetical combinations and recombinations, and when they are all again fusing to form a zygote, there is a chance of new combinations. So there will be variety. Okay, that is the advantage of sexual reproduction. So there is an opportunity for variation. All offsprings may not be alike, they will be of different types. Formation of unique combination of genetic makeup. Chance of so many different types of combination of genetic makeup will be available in sexual reproduction. Okay. And among these, so many variety of our organisms which are having different types of characters, some of them may be beneficial both to the organism as well as the population. That is the advantage. In asexual reproduction, there is no such a chance. Only one individual is involved in reproduction, and the genetic makeup of that parent will be very much given to the offspring. So, in asexual reproduction, the genetic information is preserved as such without any change. Whereas in sexual reproduction, that permits variation. That is the advantage of sexual reproduction over asexual reproduction. And we produced a large number of new organisms by cross breeding. In animal breeding, and plant breeding, we crossed organisms which are all related to each other but are all different. They may be having different qualities, and when we cross them, the hybrid or the offspring produced will be having some new combinations, new characters. Which of them will be? Some of them will be beneficial, but at the same time, it is having a disadvantage. Though sexual reproduction is having an advantage of having new varieties, production of favorable characters or desired characters, it also has a negative feature. What is that? Along with the inheritance of positive characters, there is also the chance of inheritance of undesirable characters. Along with the inheritance of desirable characters, there is chance of inheritance of undesirable characters, because we are all crossing two slightly different types of individuals in crossbreeding. So you may have a chance of inheriting good characters, but also you have a chance of inheriting unwanted, undesirable characters also. That is the disadvantage. This disadvantage is overcome by the technique called recombinant DNA technique, where we are all taking only the gene for the desired character. We are not taking the entire genome from one individual. Instead, we are all taking that gene which is responsible for our desired character only, and is giving to the organism to which you want to give that character. This is the basis of genetic engineering. So, the technique of genetic engineering include the creation of a recombinant DNA. What is a recombinant DNA? Recombinant DNA is a DNA formed by the mingling of DNA pieces from more than one source. You take DNA from one individual which may be having the gene for desirable characters, and give it to the DNA, the genome of a host organism. Now, the host organism will be having DNA pieces, one from the desirable character bearing organism, and the other one the host. That DNA, thus formally combining DNA from different sources, is called a recombinant DNA. Now, you give it to one cell and you culture them and produce large number of identical copies. That is called gene cloning. Okay, and after that, you can transfer that gene to any organism you want to have this character given to. So, you are having new techniques where you are all taking only the DNA responsible for the desirable character only. So, what is the advantage? As you are taking the DNA for the desirable character only, there is no chance of inheriting unwanted, undesirable character. That is the advantage of genetic engineering over hybridization. You got it? Then you may think it is very easy. No, it is not that easy. Why? Because how can you transfer a desirable DNA? Suppose I am introducing a small segment of our desired DNA to an organism. There are two things that may happen. One is that the enzymes, DNA's enzymes, digesting enzymes, may break it into so many pieces. Okay, this may be broken and destroyed. The host organism to which I have given the DNA, desirable DNA, that may be destroyed. That is one chance. The second chance, even if it is not destroyed, what will happen? As I am growing or culturing this host organism, it may be dividing into two. See, I have given only that piece of DNA that is responsible for a particular character. That DNA is not capable of self-division. Why it is not division or dividing? Because what I have given is only a small piece of DNA which cannot divide, start dividing. Division starts from a small segment of DNA, a small piece of DNA which is called origin of replication or ORI in brief. What I have given is not containing ORI. Where is that ORI? ORI is present in the genome, in that chromosome or in that entire DNA segment. I have not taken the entire segment of DNA. I have taken a small piece of that DNA which is having no ORI. So, because it is having no ORI, when the cell is dividing, let us see what is happening. This may be inherited to only one cell. Because it is not dividing. The cell only divides, it is not dividing. And it is not inherited to the second progeny. In the second division, only 1 out of 4. See, again it is dividing. When again it is dividing, only one cell will be getting that DNA. That means it is not inherited to progeny generations. If it is not inherited to progeny generation, the entire procedure will be end up in a failure. I want this gene to be present in all the daughter cells also, but that is not happening. So what should I do? I should mix it with a piece of DNA which is having ORI. That ORI is that piece which is responsible for the initiation of the duplication. It can duplicate. Suppose I am mixing it or adding it to a piece of DNA which is having its own ORI. Because of the presence of ORI, suppose I am giving it to a segment of DNA which is having ORI, this is having ORI, then this ORI will be dividing. When this ORI is dividing, the daughter cell will be all having. This is dividing and will be inherited. Okay, so all the daughter cells will be also getting a copy of the desired DNA. So we have to mix it with, add it with, okay, splice it with a ORI segment, and then you grow it in a medium so that you will be getting large number of identical copies, and production of that identical copies in large number is called cloning. So cloning is making multiple identical copies. Clear? Now, before we start, let us understand the basic principles. The DNA, which is responsible for a desirable character, the character I want to give to an organism, the character I want to be present in an organism, that is called desirable DNA. That DNA is called desirable DNA. DNA responsible for a desirable character. I will get it from an organism which is naturally having this gene. That organism is called donor. It is called donor. Okay. So I take the DNA, desirable DNA from a donor, and I want to give it to an organism which is called the host. Host is the organism to which I want to give, which will be receiving. So host should be capable of receiving that DNA and retaining them. It not only receives, but should sustain them and also give it to progeny generation, daughter generations, daughter cells, daughter organisms when it is multiplying. Such an organism is called host. For this purpose, I have to take the help of an agent. Our donor DNA is a small microscopic molecular piece. So we cannot see them or we cannot pick up and give it. So we need an agent. That agent is called vector. So I will take a disabled DNA and mix it with, join with the DNA of a vector. Now, the vector will be having DNA of the desirable character. For example, this is the DNA of the desirable character and this is the vector. I will cut the vector and introduce that DNA into that. Okay, I will be first cutting it and insert that disabled DNA into this vector DNA. So now it becomes okay, a hybrid DNA. It is a hybrid DNA, which is also called chimera DNA. Chimera DNA. Or a DNA that is formed by combining the desirable DNA with the vector DNA. This is called rDNA, recombinant DNA. A DNA which is formed by the combination of, by the combining of, joining of desired DNA with that of the vector DNA is called rDNA. It is that rDNA that is given to the host, so that the host will be not only maintaining them but also will pass them to the progeny generation. So this vector should have an ori. Then only this will be multiplying. When the host cell is dividing, this recombinant DNA should also divide because it is having an ori. So a suitable vector should have an ori and should multiply along with the multiplication of the host cell. That is what the principle of rDNA technology is. Now you got it? The first recombinant DNA technique, which is the most important technique in genetic engineering, was successfully conducted in 1972 by two eminent workers, namely Herbert Boyer was an expert in some enzymes he extracted from Escherichia coli. This is a cutting enzyme. We are coming to that point. And second one, Stanley Cohen, who is an expert in taking out this plasmid. What is this plasmid? Plasmid is an ideal vector. Plasmid is an extra chromosomal circular DNA present in some of the bacteria, prokaryote. Remember, bacteria are prokaryotes. Okay. Now, they will be having, a bacterium will be having their own nuclear DNA, which is larger, single circular DNA. It is a circular DNA. And in addition to that, they will be having small pieces of, small circlets of double stranded DNA. The large one is called nuclear DNA, and the smaller ones are called the plasmids. Plasmids are small pieces of circular DNA which will be containing only few number of genes. They are very small. And they are not absolutely essential for the survival of the bacterium. If a bacterium is having plasmid, the plasmid will be giving some additional qualities like ability to resist antibiotics, or such plasmids are called R plasmid, R for resistant plasmid. Cold plasmids, F plasmids, etc. You are having so many different types of plasmids based upon the quality it is giving to the bacterium. Even if you remove that plasmid, the bacterium can survive, but that additional quality a bacterium will be having will be lost because it is because of the plasmid. Such small circlets of DNA, which are double-stranded DNA, are called plasmids. Okay. So, Stanley Cohen was an expert in taking out the plasmids out of the bacterium and giving it to another bacteria. So, Herbert Boyer was an expert in cutting the DNA. With the special type of enzymes he extracted from E. coli. What is that enzyme? It is restriction enzymes. So, he was an expert on that. What they did in 1972 was that they took an antibiotic resistance gene from a suitable donor. They took antibiotic resistance gene and they gave it to the vector. What was the vector? The plasmid of Salmonella. So they took the plasmid of Salmonella, Typhimurium. That was the vector. And they gave it to a host cell which is E. coli, Escherichia coli. E. coli is a bacterium that is living as a symbiont in our intestine. Why they took this Escherichia coli as their host? Mainly because this E. coli is having naturally no antibiotic resistance ability. It cannot resist any antibiotics. That means they cannot grow in a medium in which an antibiotic is present because they cannot resist that antibiotics. Okay, but suppose you are giving a plasmid which is ligated with antibiotic gene to that E. coli. Now this E. coli is having that ability attained. Now E. coli can resist that antibiotics. That means it can now grow in an antibiotic containing medium. So simply by growing in an antibiotic containing medium, we can say that whether our recombinant DNA has successfully entered into E. coli and are working or not. If the E. coli cells are growing in the new medium containing antibiotics, our experiment is successful because the E. coli naturally cannot grow in a medium containing any antibiotics. Now it is growing. Meaning that ability to grow in a medium it obtained because we have introduced a recombinant DNA. That is the plasmid of Salmonella typhimurium plus antibiotic gene. Now that gene is working. And not only it is working, the E. coli is sustaining it, but also it is given to the daughter cells, progeny cells, because E. coli bacteria will be growing very quickly in a cultural medium. And all of them are growing means all of them are having that ability to resist antibiotics. So that is a very easy method of detecting whether our transferred gene has reached all the daughter cells also. This is the first recombinant technology that was worked out, that was achieved in 1972. Okay. So now you understand the basic principles. Now let us go to the details. Ability to multiply and produce large number of identical copies of that E. coli, which are all having the ability to resist antibiotics. So all of them are identical genetically. That is called cloning of antibiotic resistance gene. You are getting large number of copies of that gene, which are present in large number of members of a colony, which are all produced by the multiplication of the E. coli cells. That is what is called cloning. Thus, in genetic engineering, we are having three major steps. We have to identify the desirable DNA, the DNA for a desirable character. Then, introduction of that identified desirable DNA to the host cell. 3. Maintenance of the introduced DNA in the host cell. The host cell is maintaining it and transferring that DNA to the progeny cells. These are the three basic steps in genetically modifying an organism. That's all with genetic engineering. But for that, a genetic engineer should have some tools to work with. These tools are in the form of molecules, of enzymes, vectors, etc. Now let us see what are the tools. These tools are mainly of two types. One, restriction enzymes. What are these restriction enzymes? We know that we are having nuclease enzymes. Enzymes that will be cutting and digesting nuclease. We are having nuclease. In pancreas, we are having nucleases that will be cutting the DNA and RNA into nucleotides. These nucleases can be classified into exonuclease and endonuclease. What is an exonuclease? In exonuclease, the outer nucleotide, one by one, nucleotides are broken and removed because this enzyme will be attacking the ester phosphate bond of the nucleic acids that connect nucleotides together. Understand that you are having only simple diagrams. This is a nucleotide which will be having 4 different types of nitrogen bases, a sugar, pentose sugar, which may be either ribose or deoxyribose type, and a phosphate. So this is the sugar, this is the phosphate. Okay. And this is the nitrogen base. And phosphoester bond is formed between sugar and phosphate. So we call it as sugar phosphate ester bond. That's how different types of nucleotides are connected together. Okay. So this is a long chain of nucleotide, which is RNA. In DNA, you are having two strands, which are all helically coiled around each other. It is the ester phosphate bond that is broken by the enzyme. So here it is broken. Once that enzyme is broken, the bond is broken, what will happen? Nucleotides are removed from outer edges. One end will be 3 prime, the other end will be 5 prime. So from these two ends, one by one, nucleotides are removed by the action of XO. Whereas endonuclease, in endonucleases, the enzyme is cutting in the inner bonds here, so that the long DNA is cut into so many fragments. So that is why it is called endonucleases. Okay. We are having RNase enzymes, DNase enzymes, which are both exonucleases, endonucleases. Our restriction enzymes are endonucleases. Why they are called so? These nucleases were, or enzymes were, isolated by some workers like Werner Arbor, H.O. Smith, as early as in 1963. Okay. These enzymes, they studied for 5 years, and the first restricted endonuclease was extracted from a bacterium by name Haemophilus influenza RD COP. And this enzyme was called HIND2. That is the first restricted enzyme we have isolated. Okay. And that was studied for 5 years. And from that, we came to know that the HIND2 is a restriction endonuclease. What is the meaning of that term? It is an endonuclease that will be cutting, destroying a foreign DNA that entered into that bacterium. Who is entering into that bacterium? A phage, a virus, a bacteriophage. So, when a bacteria is attacked by a virus, the virus is introducing its DNA. All bacteriophages are having DNA. That DNA is a foreign DNA that is entering into a bacterium. This is the bacterium, and when a foreign DNA is entering into that foreign DNA is immediately attacked by this enzyme and is cut into so many pieces and destroyed, so that the bacteria is saved. The bacteria is having a defense mechanism, intracellular defense mechanism, just like our immunological system to protect our body from pathogens. A bacterium is having intracellular defense mechanism that will be cutting foreign DNA that is entering. Then you may ask, why it is not cutting its own DNA? See, a bacterium is having its own DNA. Why its own DNA is not cut? Because its own DNA will be having methyl groups added during its synthesis. There is another enzyme which is responsible for adding methyl group. It is methylating enzyme. That methylation occurs when its own DNA is synthesized. When is the DNA synthesized? At the time of division of the bacterium. When the bacterium is dividing, its DNA first divides first, and at that time it is methylated. So its own DNA will not be cut because it is protected by methylation. Whereas the foreign DNA that enter in is not at all having that methylation, and so that foreign DNA alone will be cut. Thus, you are having a defense mechanism in the bacterium, which is made up of two enzymes. One methylating enzyme, and the second one is restricting enzymes. These restricting enzymes are cutting the DNA on the inner bonds. So it's called endonucleases. So restriction endonucleases are a part of an enzyme complex called methylating and restricting enzyme complex. That's how a bacterium is defending itself from the attack of a virus. That was identified. And two enzymes responsible for restricting the growth of bacteriophage in Escherichia coli was isolated. One of them is adding methyl group to DNA, while the other is cutting DNA. And the second enzyme is called restriction endonucleases because it restricts, limits the growth, multiplication of a viral DNA within a bacterium. When a viral DNA enters into the bacterium, this multiplication is limited, it is destroyed by this enzyme. That is why it is called restriction endonucleases. The first restriction endonuclease was HIND2. Later on, we are having a large number of more than 900 restricted enzymes. All these restricted enzymes have been isolated from over 230 strains of bacteria. Only bacteria will be having this system restricting endonuclease enzymes because it is their defense mechanism, intracellular defense mechanism. As we are having large number of enzymes, we should have a system for naming these enzymes. And we are having a specific code for that. The first word for that enzyme, so for example, the first enzyme is HIND. Why it is called HIND? Because the first letter denotes the generic name of the bacterium from which it is extracted. I told you that the first enzyme was taken from Haemophilus influenza. So the first name is H. So we take capital letter, okay, generic name. Then two words of the specific epithet. Then D is the, small d is the strain. That is similar to subspecies, the strain. So you take that one. From the same bacterium, you would have extracted several enzymes. So what is the order in which that enzyme was extracted? If it is extracted as the first enzyme, then you can call it as 1. This enzyme was extracted as the second one, so we call it as 2. If it is a third enzyme, we call it as 3. So like that, we give that Roman letter. That is the order of the extraction of enzyme from that bacterium. So I will repeat once again. First, one letter, capital letter, that is the generic name of that species. Then 2, first alphabet of the specific epithet in small letter form. Then strain, and lastly the Roman letter indicating the order of the extraction. Similarly, eco R1, that eco denotes that it is from Escherichia. CO, coli. Eco R. R is for RY13. Okay. R is derived from the name of the strain and RY13. The first enzyme is called Eco R1, then Eco R5. Like that, you are having several enzymes extracted from that. This is how we'll be giving names for these enzymes. Clear? These enzymes are having a common property. They will be identifying a particular sequence on the DNA, which may be probably 4 nitrogen base length, 4 base, or 6 base. They will be recognizing that particular sequence, which is a palindromic sequence. For example, for Eco R1, the palindromic sequence is GAA-TTC. So, GAA-TTC. You are reading it from 5' to 3' end. A palindromic sequence is a sequence which will be the same if it is read in this strand from 5 to 3 and in the complementary strand in the same direction, that is from 5 to 3, it should be read like this: GAA-TTC. Such a sequence is called palindromic sequence. So it will first recognize a specific sequence called palindromic sequence, which is a palindromic sequence, and that sequence is called its recognition sequence. Now, after recognizing that sequence, it will cut both strands at a particular position. For example, Eco R1 is cutting always between G and A. Since it is cutting both strands between G and A, it is not in the middle, it is cutting a bit away from the middle, so that the strands, the pieces that are separated, each will be having, let us see how it is, it will be having one chain longer than the other chain. This chain is longer than the other chain. See? This type of cutting is called staggered end cutting or tail end cutting, because when it is cutting, one chain will be longer than the other one. Okay, one chain is small, the other chain is short. So the other piece will be having also one chain longer than the other one. This is what is called tail end cutting. All enzymes are not always cutting like this, but the enzymes that are mentioned here are all with staggered end cutting. Whereas some other enzymes will recognize their palindromic sequence and will cut in the middle. Say, for example, it is recognizing a sequence of the DNA and is cutting in the middle. It is in the middle. So both strands are having the same level of length. So you will be having the same length for both strands. It is called blunt end cutting. Blunt end cutting. In RNA technology, we make use of staggered end cutting enzymes. Why? Because one chain is longer than the other one. It makes the joining of DNA pieces easy. This is the reason why we take staggered end cutting. Eco R1 is a staggered end cutting enzyme whereas hint 2 is blunt end cutting. Similarly, hint 3 is a staggered end cutting, whereas eco R5 is a blunt end cutting and hint 2 is again a blunt end cutting. See, they are all obtained from the same bacterium, but their properties are different. Understand? Now, another important property of this restricted endonuclease is that it will always cut DNA taken from any source in the same way. It will recognize the same palindromic sequence and will cut in the same way. Say, for example, you take a donor DNA from a plant cell, you take a vector DNA from a bacterium, you use the same restricted enzyme. The same restricted enzyme will be cutting the same DNA, the two different types of DNA in the same way. That means all of them will be having the same complementary cut ends. This is the reason why we can easily join any piece of DNA provided both are cut with the same restriction enzyme. Herbert Boyer was an expert with this type of enzyme. He extracted from E. coli. So, ECO R1. That ECO R1 will cut a plant DNA, a viral DNA, or a fungal DNA, all with the same complementary cut ends. So, if different DNA are cut by the same restricted enzyme, they can be easily be joined together. This is the basic principle. And we should bear in mind that both the donor DNA and the vector DNA should be cut with the same restricted enzyme so that they will have the same complementary cut ends, and these complementary cut ends will be easily joined together. Okay? That is the basic principle. Same kind of sticky ends. This can be joined together end to end using another enzyme called DNA ligase. That is the second enzyme. What is that? Ligase enzymes. Ligase enzymes are enzymes that are all joining DNA pieces without a suture. Meaning, you take DNA pieces which are cut with the same restricted enzyme. You take one DNA which is cut with the restricted endonuclease, and you take another piece, another DNA, okay, which is also cut with the same restricted enzyme. They will be having the same complementary ends so that they are easily joined. We can join them correctly because of the complementary cut ends. Ligase enzymes are thus used for ligating. That's why we nickname these enzymes, restricted enzyme, as molecular scissors or scalpels for cutting. And ligase enzymes are molecular glue joining. They are just gluing. Okay. So that there is no nitrogen base lost or added. Simply, they are all joined end to end. That is the technique. Okay. Now, how you can cut a DNA and separate the DNA fragments? It is an easy technique. We are having a technique of cutting the DNA by restricting the nucleases. That long chain of DNA will be cut where there is a recognition site, and these fragments called restriction fragments, the pieces that are formed by cutting a long DNA by a restriction enzyme, are separated by keeping them in a electrical field. And since nucleic acids are negatively charged, they will be moving towards the positive. That is the basic principle. They will be moving towards the positive anode. So, in an electrophoretic apparatus, we are having a glass plate in which you are adding a medium. That medium is agarose gel. This agarose gel is obtained from red seaweeds or algae, red algae, and you are making a very dilute solution of that medium which is spread on this plate, and we add the sample that contains DNA fragment that is to be separated on the wells. Wells are in the cathode. Okay, so this is the apparatus, just basic principle. This is the agarose gel, this is the negative, this is the positive anode, and you keep a suitable electrolyte, a medium, and connect it to a battery. Then, when the circuit is closed, now what happens? The electrolyte will be slowly reaching the medium, and the DNA fragment containing solution become dissolved in that electrolyte, and that is slowly moving through the agarose gel medium according to their size through the sieving effect provided by the agarose gel. So agarose is the medium, and larger molecule will be moving slowly because it is moving by the sieving action. So larger size, slower will be the rate of movement. Smaller size, greater will be the speed at which they will be moving. So after some time, those which are reaching the positive charge anode side will be the smallest ones, and those which are in the cathode part will be the largest ones. Okay, thus we have separated the DNA pieces according to their sizes. This separation is called resolving. So, by electrophoresis, what is the meaning of that term? Electric charge bearing molecules like nucleic acids, proteins can be separated by this technique. Okay, when they are kept in a electrical medium, gel electrophoresis. The technique is called gel electrophoresis. Now, how can you see that DNA? DNA is water soluble and it is molecule. You cannot see them with your naked eyes. So, we apply a stain, which is a fluorescent stain, ethidium bromide. After that, the ethidium bromide will be combined with the nucleic acids, and since it's a fluorescent dye, you look it or expose it to UV light. Then you can see them as bright orange colored bands. These bright orange colored bands are seen. Okay, that will be the DNA fragments. Now, you understand in which piece of this DNA you contain your desirable gene. See, what you obtained is just pieces of DNA. In which piece your desirable gene is present. Suppose that your desirable gene is present here. Okay. Now, you cut it, that small piece, and keep it in a medium, a solvent. That piece is cut, that chip is cut out and kept in a suitable medium. Now the DNA get gradually dissolved, and thus the DNA is now transferred into that solution. This transferring is called elution. Now you have eluted. Okay. This is the principle in which you have isolated your desired DNA. Then, who is a vector? Vector is another important tool. So we have studied about two important enzymes, namely restricted enzymes and the ligase enzymes. Ligase enzymes are normally obtained from viruses. Okay. Now, what are cloning vectors? Vectors which are all helping as agents in transferring our desired DNA into a host. Okay. This vector should have some properties. One, this vector should be also an agent for producing large number of copies of our DNA. See, we took only one piece of our DNA, one copy of our desired gene, and that gene is given to the vector, so that in the vector it is multiplying. So multiplying vectors, multiplying and producing large number of copies is called cloning. So these vectors are called cloning vectors. Normally we use phages, viruses. Why we use phages? Phages can enter into a host cell and multiply with it. Okay. So you will be giving or you will be getting large number of identical copies, or you are having the plasmids. Plasmids are extranuclear small ringlet of DNA present in bacteria. These plasmids are also capable of entering into a bacterium and multiplying. What is the number to which it is multiplying and is present within? I will explain that point once again. Suppose I am giving this is a bacterium and I am giving only one plasmid into, and to this plasmid I have attached my desirable DNA. This is my desirable DNA. I have introduced only one or few, but after entering in, it should be capable of multiplying into large numbers. So, how much copies? Sometimes 1, 2, 3, 5, 10, like that. It will be multiplying. And to what number it is multiplying and producing identical copies? That is what is called copy number. So, what is our aim? We should get large number of identical copies of our desired gene. Okay. And suppose that each one is capable of producing 50 molecules of our enzyme in 1 minute. 50 molecules in 1 minute. I am having, say, for example, here 10 such molecules. So 10 into 50 in 1 minute. Okay. Suppose this molecule is multiplying into 100 times. So 100 are all producing 50 molecules each in 1 minute. And I will be getting more molecules of the enzyme or protein which I want to. This may be an antibiotic, it may be an enzyme, it may be a hormone, whatever may be. That is the product that I want to get it. So I will get more products in less time if I am taking a plasmid or a vector which is having high copy number. So I want to get high copy number vector. And formerly we used natural vectors like plasmids and phages because they are capable of multiplying and producing large number of copies once they enter into the host cell. But nowadays, these vectors are also genetically engineered in such a way that they will be making the easy linking of foreign DNA and selection of recombinant from non-recombinants easy. So we deliberately select that type of vectors, and the vectors that are now used are itself produced by genetic engineering. Let us see how we are all producing them. And the classical example for such a artificially prepared, genetically engineered vector is PBR322. PBR shows that it is a plasmid. That's why it is called P. The other names indicate the laboratory in which it is made. These are all genetically engineered. Bolivar and Rodriguez. Okay. And 322 is the trademark. See, they have produced a large number of such vectors. So we will be designating them by different letters or numbers to identify them. That is called the trade name. So 322 is the trade name. So PBR 322 is an ideal vector that has enabled us in the beginning of our biotechnology in understanding and perfecting the technique in biotechnology. So what are the qualities an ideal vector should have? Remember, vector is also a cloning agent. So we call it as cloning vector. It should have an ORI. Remember, ORI is that part which is capable of initiating the replication. Since a vector is having its own ORI, it can autonomously multiply without depending upon the nuclear DNA. See, this is the bacterium and this is the nuclear DNA. When I introduce a plasmid into them, since plasmid is autonomous, it is capable of self-multiplication. It need not depend upon the nuclear DNA. The nuclear DNA will multiply only when the bacterium is dividing by binary fission. But a plasmid will go on multiplying. So how many times it will multiply? That is determined by the ORI. ORI is also responsible for determining the copy number. So if you want to have high copy number, we should select a plasmid or a vector which is supporting high copy number. Clear? Then selectable marker. What is a selectable marker? It is a gene that will be helping you to select a transformant from a non-transformant. These are the terms that you should understand basically to understand this chapter. See, what is transformation and what is transformant? Now, understand it clearly. What I am giving to a bacterium, suppose this is a bacterium, say for example E. coli. I am taking E. coli as a host and I want to introduce my foreign DNA, that is recombinant DNA, into this one. See, I cannot force them into. It may enter, it may not enter. This plasmid, this is a rDNA I have constructed. It is having a plasmid plus a small segment of my desirable gene. This is the desirable gene. I want to introduce into the host cell. So this E. coli will be multiplying, and within that E. coli, this plasmid will be also multiplying into large number of copies, and I will get large amount of the products. That is my aim. So this plasmid with a disabled DNA, this is called rDNA, is to be introduced. If the whole cell receives this foreign DNA without an agent, okay, it is entering, it is taking, then it is called transformation. Suppose I am introducing a DNA with the help of a virus, an agent, then it is called transduction. Here, the DNA is taken by the host cell without any agent. This is the DNA that is entering into the host cell by itself. That is called transformation. And a cell which has taken this one, okay, now is called a transformant. Now it is a transformant. And a bacterium which has not taken, it is a non-transformant. Is that clear? Transformant and non-transformant. Now, which one you want? You want to know which bacterium has taken your recombinant DNA. So, we want to identify which is your transformant, and you don't want this one. You have to eliminate them. So, you don't want them and you want to grow this one. Selectively grow them, culture them. This alone should be growing in the medium, and this should be completely eliminated. Okay, selectively permitting the growth of transformants and eliminating the transformants. Which gene is helping, which DNA is helping in this one? That is called selectable marker. It is called selectable marker. Usually, selectable markers are antibiotic resistant genes. Okay. And in PBR 322, we are having two selectable markers. We are having two selectable markers. PST and PBU1 are resistant enzymes which are having their cutting site in ampicillin resistance. That is a selectable marker. Again, SAL1, Salmonella albus, and BAM-H1, Bacillus amyloliquefaciens, H1. They are having the cutting site in the second selectable marker, namely tetracycline resistance. These are all antibiotic resistant genes. So normally, antibiotic resistant genes are taken as selectable markers. Selectable markers are those genes which are all helping in identifying and eliminating non-transformants. That is, those who are not taken, and selectively permitting the growth of transformants. Such genes are called selectable markers. Clear? Then, the third important quality is that they should have cloning site. What is a cloning site? See, cloning site is a site. This is the plasmid, which is our vector. Suppose I am using a restricted enzyme, and that restricted enzyme is having so many cutting sites. It is cutting here, it is cutting here, like that. It is cutting into so many pieces. Then what will happen? The vector will be, the DNA will be cut into so many pieces, and I cannot make it back. Some piece may be lost when they are all again rejoined. So there will be a lot of difficulty if that vector is having more than one cutting site for a particular enzyme. So what I need, it should have only one cutting site. Very few preferably only one cutting site. It is cutting only here. Okay. So that only in that cut site, a foreign DNA will get attached to. That attachment of a foreign DNA to this DNA, that joining is called ligation or splicing. Okay, so my foreign DNA, that is the desirable DNA, I am wishing to have that DNA come and joined here. It will be cut here only at the particular palindromic sequence only, and so my desirable DNA will be coming and joining at this site only. Clear? It cannot come and join in any other site. It can come only where a wound is created, a cut is created. So I have spliced it to this site. And it is this site that is multiplying. It is this DNA that is multiplied into a number of copies. So we call it as cloning site. This is the site where I have added my desirable DNA, and so now this is the desirable DNA that is coming and joining, that is cloning. So the cutting site is also the cloning site. So cloning site. So our vector should have preferably one single recognition site for the commonly used restriction enzymes. PBR is having 4 restricted enzymes which will be cutting within the selectable marker. We should use a restricted enzyme which is having their cutting site, only one cutting site within the selectable marker. That is the technique. So what is happening? Suppose I am using BAM-H1 enzyme. Understand? I am using BAM-H1 enzyme. So what will be the, this will be cutting here. So my foreign DNA comes and binds here. Right? When that foreign DNA comes and binds here, what happens to the tetracycline gene? This gene becomes inactivated. Why? Because the gene is cut and foreign DNA is inserted within. So the structure of that gene changed, and so the tetracycline gene is not functioning properly. It will not work. That is my principle. Okay. So the basic principle is that I should use a vector which is having preferably one recognition site, that is one cloning site, one cutting site for a restriction site. PBR is having 4 restriction enzymes cutting site. BAM-H1, SAL1 in tetracycline, PVU and PST in ampicillin. Here you are having a rope. What is rope? Rope is proteins for replication. Since a vector is an autonomous, it should code for all the proteins, enzymes that are needed for its own replication. It need not depend upon the host DNA. That is what is called rope. And this is the ORI. Suppose ORI is having a cutting site, we cannot use it. So it should not have a cutting site in ORI. So a total of 8 restricted enzymes are present in PBR 322, but we can use either these 2 or these 2, only one at a time. Suppose I am starting with my experiment here. It is cutting here and the foreign DNA will be inserted. Now, what will be my technique? I want to know which is a transformant and which is not a transformant. For that, I will be using some techniques. Okay, that is what we are now going to study. In modern times, in addition to these artificial plasmids, we are also using some viruses or some dangerous bacteria like Agrobacterium tumefaciens. This Agrobacterium tumefaciens is a soil living bacterium which is capable of producing a disease called crown gall disease, a tumor, cancer. This bacterium is a dangerous one. It is having its own nuclear DNA, but in addition to that, it is also having a plasmid. That plasmid is called, the TI plasmid. I told you that plasmids are given different names. TA, tumor inducing plasmid. They will be causing tumor, cancerous growth. This is a dangerous one. How it is working? The TA plasmid is having a small piece of DNA within it. That piece of DNA is called tDNA. It is this tDNA that is given to the root and it is given to the cells of the dicot plants and it causes tumor. Okay, so we have studied it. It is a genetical engineer which is capable of transferring a part of its TA plasmid called tDNA into the host cell. Once it is given to the host cell transforms itself into a tumor, crown gall tumor. So we understood that. So what we did is that we took that plasmid out. Cut and removed the tDNA and that was replaced by our desirable DNA. The tDNA is removed and is replaced by our desirable gene and give it to the dicot plant cells. So this desirable gene will be given to the genome of the plant cell. That is a wonderful discovery. Agrobacterium tumefaciens is called nature's or natural genetic engineer. So this is a pathogen which is now made our friend. We have disarmed that pathogen. The technique is called disarmed pathogen technique. Similarly, retroviruses. Retroviruses are capable of producing a DNA from its RNA and that double-stranded DNA produced will be incorporated into the genome of the host animal cell. Okay, they will be thus transferred and given to the genome of the animal cell. So we understood the technique. We take that very harmful retroviruses and that viral genome is removed and is replaced by our healthy gene, normal gene which we want to give and then infect it to animal cells. So what will be the result? Our desired gene will be given to the animal cells. Thus, plants and animals are now given our desired gene using this disarmed pathogen technique. Agrobacterium tumefaciens for plant cells, dicot plant cells, and retroviruses for animal cells. Clear? Now, how DNA will be entering into, naturally, no cell will allow the DNA to enter in because DNA is water soluble. Plasma membrane will allow only lipid soluble material to enter in. So DNA is not allowed. So we have to persuade the DNA to enter into the host or persuade the host cell to take up the DNA from outside. That is what is called making them competent. We have to make them competent. How we will be making them competent? They are all made competent by several mechanisms. One method for bacteria, especially used for bacteria, is heat shock method. You keep the bacterium after treating with a divalent cation like calcium. Okay, the bacterium cells are treated with calcium That is called treating with a divalent cation like calcium, and after that keeping it in ice. From ice, it is suddenly changed to 42 degrees and again brought back to ice. Again 42, again ice. Thus, after performing this type of action, there may be sometimes small pores produced. Through these pores, small pores, okay, the size of the pore I have made is very big. Don't think like that because the screen is like that. I cannot make smaller ones, smaller dashes. So through these temporary pores, a DNA can enter. This is what is called heat shock method. Okay, that is a common method. Another method that is for plant cells is biolistic or gene gun method. It is called gene gun method. It is for plant cells. Here I am using a colloid of gold or tungsten. Okay. This fine microscopic colloid of gold or tungsten is now coated with the DNA. Now I am coating them with the DNA, that desired DNA. And this is the bullet. And with the help of a gun, I am targeting, okay, cells. And with the help of the gene gun called biolistic method, it is also called biolistic method. I am targeting plant cells. Plant cells are aimed and this will be entering into. So our desired DNA will be entering into the plant cells. Okay. Now, if it is animal cells, you can have micro injection. That is, you take that animal cell and inject directly the foreign DNA using a micro pipette or syringe under microscope. This can be applied only for animal cells. Okay, so you can use micro injection technique. You can also have electrophoresis. What is electrophoresis? Here you are keeping it in a chamber, an electrical chamber. The cells are given electric shock at high voltage so that they will be having temporary pores produced. These temporary pores will be helping them temporary pores will be helping them in taking the foreign DNA into them. So through these temporary pores, they may enter in. Okay. So electrophoresis. So these are the techniques by which we are all compelling. That is called competency. We are making them competent to take generally by heat shock. Gene gun method, micro injection, and electrophoresis are vectorless transfer. That is direct transfer. Without an agent, we are all directly transferring, giving our desired DNA into a host cell. Okay. So, competent host for the transformation. Now, what is the procedure? After finishing the discussion, we are going to the Now we go to the procedure. What is the procedure? First of all, we have to isolate the cell from which you want to get the desirable G. It may be anything. So for example, it may be a plant cell. If it's a plant cell, first you have to cut the plant tissue into pieces and mix and make a homogeneous solution, and that solution should be treated with cellulase enzyme. That will be bursting, that will be digesting cellulose cell wall, so that the cell will be releasing the nucleus. Okay. If it is animal cell, there is no cell wall, so there is no such problem. You simply cut it and make it a homogeneous solution. And if it is fungus cells, it is to be treated with chitinase. If it is bacteria, you treat it with lysozymes. Okay. So the cell wall is broken. After cell wall is broken, you treat it with, or you subject it to ultracentrifugation. You perform ultracentrifugation. So high-speed centrifugation will be causing the cells to burst and release the inner contents. So from that, we have to remove one by one the ingredients, which are all heavy molecules. First, treat it with protease. So protein will be all broken down into small amino acids. They become lightweight, so they will come to the surface. You remove that. Okay, like that, you remove all the carbohydrate by treating with amylase, lipase, and finally RNase. So all other molecules, heavy molecules are removed. What is remaining is only the DNA. That DNA will be purified and you add chilled ethanol, so that the DNA now precipitated in the form of long threads. That is taken out. That is called spooling. It is called spooling. Now you got the DNA from a cell. Now what should be the next step? You have to feed that DNA with restriction enzyme, which you have selected. That is called restriction enzyme digestion. So the DNA will be cut into so many pieces. Wherever you are having the recognition site, the enzyme will join there and will cut into so many pieces. Okay. These pieces are then subjected to what is called resolution. They are to be resolved in agarose gel electrophoresis. So you will be having the DNA separated based upon their molecular size. Then you isolate your piece of DNA having your desired DNA, and it is transferred into a suitable solvent, that is called, you have eluted elution. Thus you have transferred that DNA. That DNA, that is desired DNA, you obtained with much difficulty. Now you are having a technique of making large number of copies of that desired DNA. Okay. Similarly, you take the plasmid vector or the vector as the case may be. Treat both of them with the same restriction enzyme. This should be also cut with the same enzyme that you are used for cutting your desired DNA. And these two pieces, that is your desired DNA piece and this vector that you have cut. Okay, you have cut it. Now you have cut it. They should be mixed together. They should be mixed together by adding ligase enzyme. They have to be added. The ligase enzyme. So when you are adding ligase enzyme, you will be having three possibilities. Sometimes the vector is not at all cut. Vector will be uncut. So it cannot take any foreign DNA. Sometimes vector is cut and the foreign DNA might have entered into. Okay. We don't know whether it has happened or not, how much vector had taken the DNA, foreign DNA, and how much has not taken it. Now, you give it to the treated competent host cell. What is that host cell? E. coli. So you add E. coli cells and add this mixture. Treated host cell to that mixture. So what will be happening? There are three possibilities. There are three possibilities. One, the foreign DNA has not entered in. After treating with the host, you can have three types of host cells. Number one, no DNA is taken by this one. So we call it as non-transformant. It has not taken anything from outside. Two, transformants. These transformants are of two types. It is of two types. Transformant is of two types. One, it has taken only the plasmid. This plasmid alone had entered. It is called non-recombinant transformant. Whereas the three is the recombinant transformant. It has taken a recombinant plasmid. So a recombinant plasmid entered in. So you can have three types of host cells. Okay. Now, how you will be recognizing them? This recognition is based upon a simple technique of culturing, plating in two different media. This culturing in two different media is called selective it is based upon culturing in two separate media. Let us see what these media are. Suppose you are using, we started our experiment with BAM H1. So BAM H1 cut and the foreign DNA will be coming in the tetracycline. So you first culture them in the media containing ampicillin. Then you culture it in tetracycline containing antibiotic containing medium. You will be having all the three types. 1, 2, 3. All the three types of bacteria. Let us see what is happening. What is number one? Number one is non-transformant. It has not taken anything from outside. Since it is non-transformant, it cannot grow. Because E. coli is having no ability to resist any antibiotic. It has not taken the plasmid from outside, so it cannot grow. Now, what is 2? 2 is a transformant. What type of transformant? It has taken only a plasmid. That plasmid is giving the ability to resist ampicillin, so it will grow. It will grow. But What about the three? The third possibility is that this is growing. See, this having the foreign DNA, it is the recombinant transformant. This recombinant transformant will also grow because it is having the amphicyclic resistant gene correctly in the position intact. So it will also grow. So who is not growing is the non-transformant. So non-transformants are eliminated and only transformants will grow. Now in the second culture medium, you have the same pattern. Non-transformant, since it has not taken anything from outside, will not grow here. So it will be dying. It cannot grow because it is a non-transformant. What is a transformant? Transformant will be growing here because it is having both antibiotics, amphicyclic and tetracyclic genes in position incorrect without any damage. So this will grow. And what about the three? The three. Second one is non-recombinant transformant. And third one is the recombinant transformant. They cannot grow. Why? Because in them, the foreign DNA is inserted within the tetracycline gene. So tetracycline resistant gene is damaged. It is damaged. So by the insertion of a foreign DNA, it has become inactive, functionless. So it cannot grow. So which one is your recombinant transformant? This is the three. That is growing in the first medium but not growing in the second medium. Non-transformant will never grow in both media. Non-recombinant transformant will grow in both medium, and our required recombinant transformant grows only in the first medium but not in the second medium. So this technique, when we are using PBR, is a difficult one because we have to do simultaneously two plating, two cultural media is to be prepared and bacteria is to be grown in these two. And still our required recombinant transformant is growing on the first medium only, where it is growing along with the non-recombinant transformants also. So to make them distinct, we are all selecting, or we are all again doing a plating in a second medium containing tetracycline where it is not growing. So we have developed another technique, that is using a coloring gene that will be producing. That is what is done. But for that we have to use another artificial plasmid like PUC. That name is not in our books. Plasmid developed by the University of California. This is having a gene called lakhs gene that is responsible for the production of an enzyme, beta galactosidase. This beta galactosidase enzyme is produced by the lakhs gene. Okay. And this is having only one antibiotic resistant gene, amphicyclic. And it will be having a large number of cutting site for different restriction enzymes. So whenever that restriction enzyme will be coming and cutting, the lakhs gene cannot produce this beta galactosidase enzyme. This beta galactosidase enzyme cannot be produced because it is very close to the lakhs gene. That lakhs gene is obtained from our E. coli. Okay. Now, this PUC is having a chromogenic. We use a chromogenic substrate. What is that chromogenic substrate? In this, we are using a cultural medium that will be having ampicillin and also a chromogenic substance, a coloring agent, chromogenic substrate on which the beta galactosidase enzyme will be working. Suppose you are transferring all the three types of bacteria into this cultural medium. So, for example, the same one, 1, 2, 3. What is one? It is a non-transformant. Non-transformants will not grow because here it is having ampicillin in the culture medium. E. coli has not taken anything from outside. So this E. coli, this type of non-recombinant cannot grow. So they will be eliminated. Second one is non-recombinant transformant. It has taken only a plasmid from outside. It has taken a plasmid from outside. And since it is having a plasmid, it is having ampicillin resistant gene. And since it is having lakhs gene, it produces the enzyme beta galactosidase. This beta galactosidase will be coming out and will be converting this chromogenic substance into blue colored product, blue products. That is why we use that chromogenic substance, blue colored products. So this will grow as colony with blue color. Whereas the third one, what is our third one which is having our desired DNA? We don't know whether it has taken it or not. We are trying to select it out to identify. This is a recombinant transformant. This will also grow because here it is the cutting site. So ampicillin gene is not at all affected. So it is having ampicillin gene intact and so it will be also growing. But since foreign DNA has come and inserted with the lakhs gene is not producing enzyme, not producing any enzyme, no enzyme. Since no enzyme is produced, there is no color. So they will be growing as white colored colony. So you can select those which are growing as white colonies, which are all having our recombinant transformant. Our desired DNA is now present in this type of host cells. We can eliminate all this. This will never grow. So there is no problem of elimination. And we can eliminate because of the blue color. We can easily find it in the cultural medium and eliminate all that colonies. So this has become more easy nowadays. Is that clear? So we are having a problem with this one. Thus, after having successfully done this technique, now we can use another technique for getting large number of copies of our desired DNA. See, it is after a long experiment that you got one single copy of our desired DNA. Now you can have large copies produced. How? How you are all capable of producing large number of copies? That is with a simple instrument called PCR. It is by an instrument called PCR. Polymer chain reaction. PCR is a technique, it's an instrument actually, where we are all using polymer chain reaction technique to get maximum number of copies of our desired DNA. Here, there are 3 steps in which you are all having the actions occurring. In this automatic machine, which is called thermocycler, we are all adjusting and making all the ingredients present. 4 types of materials we have to add. One is the template DNA, that is the DNA that we want to get amplified. We want to get large number of copies of that one. 2, all the 4 types of deoxyribose triphosphates of ATP, GTP, CTP, TTP. 3, a polymerase enzyme that can withstand high temperature up to 92 degrees. That is obtained from a thermophilus bacterium. So we are having a special type of Taq polymerase, Thermophilus aquaticus. From that bacterium you are taking, this is a bacterium that is capable of living in hot springs. So you take that enzyme and first one, large copies of two types of primers, small segments of DNA which are artificially prepared. So these four things you have to add along with the buffering agents. Then start the machine. Initially, the temperature will be going to 94 degrees. During that, the two strands of the DNA separate. This is called the denaturing of DNA. DNA become separated. Now, the temperature is automatically coming to 50 to 60 degrees. That is the second step, during which primers get attached to the single-stranded DNAs. So primers are annealed, primer annealing. It is called primer annealing. And immediately the temperature is again raised. That is the third and the final stage, where the temperature is raised to 72 degrees. It is the ideal temperature in which Taq polymerase are acting. So when the Taq polymerases are acting, the polymerization will go on, that is chain elongation. So at the end of one cycle, the two strands are separated and two new chains are produced. So you will be getting two molecules. At the end of the second, it becomes 4, then 8, like that. So at the end of 10 cycle, an average of 1000 molecules of DNA will be obtained. And at the end of 20 cycles, 1000, that is 1 million. At the end of 30 cycles, 1000 million, that is 1 billion. At the end of 40 cycles, 1 trillion. Like that, you will be getting a large number of molecules of a desired DNA. This technique is called PCR, discovered by Carey Mullis. Okay. Now, after making this discovery successful in the laboratory, you grow them, your transformed DNA, and see whether they are all producing your desired DNA or not. That desired DNA will be produced only in small quantity, but you see, verify yourself that it is working or not. Correct, it is working. When you are satisfied with the products, then you have to give it to an industrial dimension. You have to produce it in large quantities. Okay. Now, your DNA is working in a different host. You have taken a DNA from one host, one donor, and given to a host cell, which is different. This is called a recombinant protein. What is a recombinant protein? Protein produced in a heterologous host cell is a recombinant protein. That recombinant protein you want to produce in large bulk quantities, for which we can use a bioreactor. A bioreactor is a huge vessel, okay, in which thousands of liters of cultural medium can be added and culture them. The bioreactor will be having all the favorable conditions needed for the optimum, maximum growth and multiplication of the host cell. Maybe a bacterium cell, a plant cell, or animal cell. You want to culture in large numbers, so that you will be getting the product in bulk quantities. For that, you have to keep the cell growing in the maximum log phase. For that, you give all the conditions favorable. Such a huge chamber is called a bioreactor. There are two types of bioreactors: stirred tank bioreactor and sparging. In stirred bioreactor, there is a system for stirring, so that the content are mixed thoroughly. The content will be mixed thoroughly, so that oxygen and other nutrients are reaching every cell, and they will be growing. And they secrete their proteins to the cultural medium. You take that cultural medium from the sampling port, a port when you are taking out the used cultural medium and add fresh cultural medium again. So you are not stopping the reactions. Reaction is always going on at an experimental or logarithmic phase, and your product will be available in maximum amount. Okay. Whereas in sparging type, the oxygen molecules are sparged from the bottom so that oxygen become very easily available to every part. Okay. So these are the two types of bioreactors that are available nowadays. Now, after taking out the used cultural medium, you have to separate that recombinant protein from that used cultural medium. The overall procedure is called downstream processing. It is called downstream processing. For that, you have to isolate that protein, purify them, standardize them, add suitable preservatives, and standardize them. See, what is the strength of that protein in 1 unit of that or 1 ml of that solution? Like that, you have to standardize. Then do clinical trials, then field trials. Give it to the experimental animals. See what the results are. Analyze the result. If it is encouraging, you can give it to human beings. If it is to be given to the human beings. And thus you make trials before you market. The entire process, right from taking out the used cultural medium to marketing, is called downstreaming process. See, what is our ultimate aim? We have to get that product in bulk quantities by genetic engineering. We made a plant cell or an animal cell or a fungal cell or a bacterial cell to produce that product for us. That was our aim of genetic engineering, and we have achieved it. And with this, we conclude the discussion of this chapter, and we'll meet with another chapter next time. Until then, goodbye, all of you. So thank you all. Thank you very much.